The Golgi apparatus, also known as the Golgi complex or Golgi body, is a vital organelle in eukaryotic cells. It acts as a processing and packaging center for proteins and lipids synthesized in the endoplasmic reticulum, directing them to their final destinations. Parts of the Golgi apparatus. The Golgi apparatus has a distinct structure consisting of three main components. Cisternae, these are flattened, membrane-bound sacs or disc-shaped compartments arranged in stacks. A typical Golgi stack contains 4 to 8 cisternae, although some organisms can have up to 60. The cisternae are held together by a proteinaceous matrix and supported by cytoplasmic microtubules. Each cisterna encloses a lumen, which contains enzymes and fluids necessary for modifying and sorting molecules. Tubules. These are short, branched, interconnected membrane structures that extend from the cisternae, particularly at the periphery and the trans face, which is the exit side of the Golgi. They form a network and are involved in the final stages of protein modification and packaging. Vesicles. These are small, membrane-bound sacs that bud off from the tubules and the trans face of the Golgi. They serve as transport containers, carrying modified proteins and lipids to their designated locations within or outside the cell. Vesicles can be coated, like with proteins such as clathrin, or smooth, depending on their function. Some vesicles mature into larger Golgian. The Golgi apparatus exhibits structural and functional polarity, with distinct entry and exit faces. Cis face, also known as the forming face. This convex side is oriented towards the endoplasmic reticulum and is where transport vesicles carrying newly synthesized proteins and lipids arrive and fuse with the Golgi membrane. Trans face, also known as the maturing face. This concave side faces the plasma membrane and is where the processed and packaged molecules are sorted and dispatched in vesicles to their final destinations. Medial cisternae. These lie between the CIS and trans faces and are involved in various stages of protein modification. Cis-Golgi network, or CGN, a network of interconnected tubules and vesicles associated with the CIS face, responsible for receiving cargo from the ER. Trans-Golgi network, or TGN, a network of interconnected tubules and vesicles associated with the trans face, serving as a sorting and packaging station for molecules destined for different locations. So, the Golgi membranes are primarily composed of phospholipids, proteins, and carbohydrates. The lipid composition differs slightly across the cisternae, reflecting the different enzymatic activities within each compartment. A diverse array of enzymes resides within the Golgi, each responsible for specific modifications of proteins and lipids. These include glycosyltransferases for adding carbohydrate chains, glycosidases for removing carbohydrate chains, kinases for phosphorylation, and sulfotransferases for sulfation. Matrix proteins provide structural support. Various carbohydrates such as glucose, galactose, mannose, N-acetylglucosamine, and sialic acid in animal cells are present as they are added to proteins and lipids during processing. Plant Golgi lacks sialic acid but contains other carbohydrates like xylose and arabinose, reflecting the Golgi apparatus plays a crucial role in several cellular processes. Protein modification and sorting. This is a primary function. Proteins entering the Golgi from the ER undergo a series of post-translational modifications as they move through the cisternae. These modifications include glycosylation, addition of carbohydrates, phosphorylation, sulfation, and proteolytic cleavage. The Golgi also sorts these modified proteins according to their final destinations, for example, lysosomes, plasma membrane, secretion. Lipid metabolism. The Golgi is involved in the synthesis of certain lipids, including glycolipids and sphingomyelin. It also modifies lipids received from the ER. Polysaccharide synthesis in plant cells. In plant cells, the Golgi is the primary site for synthesizing complex polysaccharides that form the cell wall. Vesicle formation and transport. The Golgi packages the modified proteins and lipids into transport vesicles that bud off from the trans-Golgi network. These vesicles then deliver their contents to specific locations within or outside the cell through various transport pathways, for example, constitutive secretion, regulated secretion, delivery to lysosomes. Lysosome formation. The Golgi plays a role in forming lysosomes by packaging hydrolytic enzymes into vesicles that eventually fuse with late endosomes to become lysosomes.